from around the world, wherever you're joining from around the world, to this webinar on heat treatment of milk, an overview of technologies, results and consequences of treatment. This webinar is being sponsored and hosted by the International Dairy Federation. My name is Jeffrey Smithers. I'm joining from Melbourne, Australia, and together with Aurélie Dubois at IDF, we will be moderating today's webinar. The purpose of today's webinar, I guess, is twofold. The first is to promote a recent publication, a bulletin of the IDF that addresses heat treatment of milk. IDF has generously provided this publication free of charge, and it can be downloaded from the IDF website. Simply go to the IDF homepage, click on the publications link at the top of the page, and it should appear in the list of publications, freely downloadable. The second purpose of the webinar is to highlight a number of aspects addressed in this publication. And for that purpose, we have 4 experts with us today who will make presentations shortly on various aspects covered in the IDF bulletin. I would encourage you to ask questions, and to do that, please use the chat mode at the bottom of the Zoom page. And we'll try and address as many of those questions in the question and answer session at the end of the 4 presentations. If you could also remember to please mute your audio while our presenters are presenting. So without further ado, let me introduce our first speaker, someone from Australia who I know very well. Professor Hilton Deith. Hilton has extensive experience in dairy research involving milk and a range of dairy products, and he specialised over his career in thermal processing. He joined the University of Queensland in 1995, where he's taught food science and technology, and from 1996 to 2008, he was director of the Australian Centre for UHT Processing at the University of Queensland. He's published extensively with more than 160 research papers and 40 chapters, and he's also co-authored what is considered a seminal publication in the topic area of today's webinar, a book entitled High Temperature Processing of Milk and Milk Products. Hilton is now an emeritus professor at the University of Queensland, and he consults to the dairy industry. With that brief introduction, let me hand over to Hilton, who is going to address the topic of heat treatments of milk and consequences of their treatment. Over to you, Hilton.
Thanks, Jeff. How do I get up? Is someone going to put me on there, or?
Yes, you should show. I will unpin Jeff.
Just share your screen, Hilton.
Yeah, I can do that.
There we go. Okay. If you go into presentation mode.
Right.
There we go.
Okay.
Over to you.
Thanks, Jeff. Thanks for the introduction, and welcome to everybody to this webinar. What I want to speak about today is the heat treatments of milk, and look at some of the consequences of these heat treatments. By way of outline, I'll spend a little bit of time on thermal processing, then a bit on bacterial destruction, enzyme inactivation, chemical changes, and their consequences will be the major part of my presentation. So in terms of background, oops, go back one. As we all know, thermal processing is the most widely used technology in the dairy industry. we do now have quite a lot of non-thermal technologies, but none of those have been nearly as successful as thermal processing up until this time. Thermal processing was initially introduced to preserve drinking milk by killing spoilage bacteria, and I guess that's the process we now know as pasteurization. But over time, a large range of heat treatments have been introduced to the dairy industry, and I just want to talk a little bit about those, and I guess that's the basis of the IDF bulletin. The bulletin begins with a table which summarises the various heat treatments, commencing with the mildest, which is thermization, and then going on to the more intense heat treatments, as well as some specialised treatments for production of powders and yogurt. And I've just put up here the beginning of that table, which is titled Major Heat Treatments Used in the Dairy Sector. And this one looks at thermization, and the table looks at applications of the various heat treatments, the bacteria destroyed, significant chemical effects, and a few comments. So it really is a summary of the whole bulletin in many ways. So the major, not sure why I've got a little thing up there. The major reasons for heat treatments are obviously to kill bacteria, but also to inactivate enzymes and to produce certain physical effects. And these are caused by heat-induced chemical reactions. And I want to talk about a few of those. So I just want to talk about some of the bacteria targets of heat treatments. And I've listed the different heat treatments here, from thermization through to in-container sterilisation. I've put the approximate heat treatments in there. They're very approximate. Obviously there's a range of temperatures and times used in the various treatments, but I've just put an approximation for these here. So thermization was introduced to reduce the levels of spoilage bacteria, and we're going to hear more about that in another presentation. Pasteurization, and that the target there is the most heat-resistant, non-spore forming pathogenic bacteria, and that's Coxiella burnettii, the one that causes Q fever. It also, that pasteurization condition also inactivates Mycobacterium tuberculosis, which used to be the target, but Coxiella is slightly more heat resistant. And then extended shelf life processing, the process that comes between pasteurization and UHT processing, for which there's no formal time temperature combinations that have been made. And then that's to, the target there is to kill spores of psychotrophic spore forming bacteria, especially Bacillus cereus, and we're going to hear about that more later on. And the reason why the spores of psychotrophic spore forming bacteria, the target there, is because ESL milk is kept at refrigeration temperature. Then UHT processing, the targets there are the spores of most spore forming bacteria. There's a few very heat resistant spores which still are not inactivated by UHT processing. And then in container sterilisation, basically to kill all the bacteria, but significantly to cause a 12-log reduction in the spores of Clostridium botulinum, the so-called botulinum cook. So, whoops, okay. So the enzymes that are of interest, as we know, there's a lot of indigenous enzymes in milk. I think over 60 have been identified, but only a few of these have significance in relation to heat treatments. And I've just listed 3 here. Alkaline phosphatase, which is inactivated by pasteurization. and used as an indicator of the adequacy of pasteurization. It's interesting to note that thermized milk contains active phosphatase, so it's got a positive phosphatase test. Lipoprotein lipase, it's inactivated by pasteurization, which is fortunate because otherwise all of our homogenised pasteurised milk would be rancid, and that's obviously not the case. And plasmin, a very heat-resistant alkaline phosphatase, alkaline protease. It's inactivated by very high intensity UHT processing, and certain preheat processes that are used in UHT processing. In general, it's not inactivated by most direct processing unless they have a fairly intense preheat section. So I want to get on to the heat-induced chemical changes, and here I'm not going to talk much about thermization and pasteurization because they cause a little chemical change, and by little chemical change I mean the changes that result in colour, flavour and texture alterations. So the noticeable changes occur with the higher temperature heating, and I've nominated 90 degrees as the temperature above which I want to talk about the changes. And the 5 changes I want to talk about are denaturation of whey proteins, the Maillard reaction, cross-linking of proteins, lactose isomerization, and vitamin destruction. And for each of these, I want to talk about what happens and then talk about the consequences of those changes. So in terms of denaturation of whey proteins, I guess I'm mainly concentrating here on beta-lactoglobulin. It's not the only whey protein, but it is the major one, and it has the major effect in terms of denaturation. So the unfolding of beta-lactoglobulin exposes free sulfhydryl groups and also some hydrophobic parts. These unfolded whey proteins then interact with each other and with caseins, and they form protein-protein interactions via sulfhydryl disulfide interactions, as well as hydrophobic interactions. And I guess some of the consequences of that is that the sulfhydryl groups form during whole milk manufacture, for example, which is 9 degrees for about 30 seconds. And that has an antioxidative effect and reduces oxidation during storage. So that particular time temperature combination has been chosen to produce an antioxidant effect. The protein-protein interactions, they have numerous effects in numerous compounds and numerous products, but I'll just mention a couple here. The increase in viscosity of yogurt is largely due to the heat treatment that the yogurt mix gets, and that's a major reason for that time temperature combination. And also the whitening of milk. Once you have an interaction between whey proteins and the casein, there's an increase in reflectance of light. The sulfur amino acids are also degraded during the denaturation process to form volatile sulfur compounds, and these are well known: hydrogen sulfide, methane thiol, dimethyl sulfide, and dimethyl disulfide. And I guess the consequence of that is that we get a heated cooked flavour in products, particularly UHT milk. And of course, the unfolding of beta-lactoglobulin increases the access of proteolytic enzymes, and that has a consequence of increasing the rate of digestion. Now, of course, denaturation increases with the severity of heating, and that's taken into account in the classification of skim milk powders, which is based on the whey protein nitrogen index, which is the amount of undernatured whey proteins per gram of powder. So your low heat powder has to have more than 6 milligrams of undernatured whey proteins per gram of powder, and then right through to the very high heat powder, which has much less than 1.5. Now, denaturation of whey proteins is sometimes used as an index of the heat severity in UHT processing, but it's not a particularly good measure because if you have a very intense preheat treatment, but it can result in a low amount of undonatured whey protein, but the whole process may not be very severe. The second one I want to talk about is the Maillard reaction, and that occurs in 3 stages, and I've put that there purposely because each of those has some effect in some products. So the early stage is the reaction of lactose with amino acids, particularly lysine. It reacts with other amino acids as well, but mainly lysine. And remember, these are in proteins, so they're protein-bound interactions. The first stable product is the protein-bound lactulose lysine. So the lactose has been isomerized to lactulose there. The intermediate stage, mostly small molecules are formed there, hydroxymethylfurfural, dicarbonyl compounds, and various flavor compounds. And then, of course, the final stage is where we get melanoidins, the brown pigments. And I guess a lot of us are used to calling it Maillard browning, and that's probably the first thing we think of with Maillard reaction. And in that final stage, there's also advanced glycation end products being formed. And that's what these are now termed, AGEs, particularly carboxymethyl lysine. So some of the consequences of these reactions: the lactulose lysine forms furacine when we digest it with acid. Just note that furacine is not present in the product itself, as despite what a lot of papers do say. Furacine is used very widely as an index of severity of heating and is a very good index for freshly manufactured products, but not for stored products as lactulose lysine and the Maillard reaction continues during storage. The lysine, of course, in lactulose lysine is blocked, which means that it's unavailable nutritionally. This is significant for some products and particularly for infant formula. The intermediate stage, the dicarbonyl reaction products, things like glyoxal and methylglyoxal, they've got 2 carbonyl groups which are ideally placed to link proteins, and so you get cross-linking, and this cross-linking can contribute to the loss of solubility of products, particularly powders such as milk protein concentrate. Hydroxymethylfurfural, sometimes used as a chemical index for severity of heating. Again, not particularly good one, because during storage of product, HMF is both formed and degraded. And then, of course, the flavour compounds formed in the Maillard reaction are pretty important in terms of the flavour of heated milk, such as UHT milk. Some of the compounds that contribute to that flavour are maltol, diacetyl, and furfural. And in the final stage, I mentioned the glycation end products. And the reason for mentioning those is that they've been implicated in diseases such as diabetes, and the medical profession has taken on that term. We used to call them advanced Maillard reaction products, but now they're being called glycation end products. The reason for the interest is that they've been implicated in diseases such as diabetes. However, the significance of the dietary AGEs is still somewhat unclear. Of course, the highly heated milks develop a brown coloration. I've just put up a photo there of milk jam, or dulce de leche, whatever you like to call it, heated condensed milk. Very popular product in South America, of course. A little bit on protein cross-linking. I've mentioned it already in terms of Maillard reaction products, but it can also occur through the reaction with dehydroalanine and lysine. That forms the isodipeptide lysinoalanine. Now, dehydroalanine is formed during the heat process by elimination of water, H2S or phosphate from serine, cysteine, or phosphoserine. The production of LAL increases with the pH of heating. So if we've got a product with a high pH, we're more likely to get a higher level. Some of the consequences, the formation of LAL obviously reduces the amount of nutritionally available lysine. And dietary LAL has been linked to abnormal kidney cell growth in experimental animals. So we really want to keep that to a minimum. The levels in heated milk at normal pHs are really not a health hazard, but they can be high in some products at high pH. And I guess caseinates that are formed at high pH are one of the ones that have been implicated. LAL used to be a problem, particularly in roller dried powders, where the heating is quite severe. But of course, we don't use roller drying very much these days. Lactose isomerization, just quickly, that's a conversion of lactose to lactulose by the heat. So lactulose is a disaccharide, galactose fructose, compared with lactose as galactose glucose. The levels in increase with severity of heating. So I guess the consequence of that is that it's been used as a chemical heat index, and it's probably the best one we've got. And the reason why it's the best one we've got is that it's not present in raw milk. It increases with severity of heating, and it doesn't change during the storage of processed milk, or it changes very little. So it's probably the best index of chemical, the best chemical heat severity index that we've got. And lastly, vitamin destruction. And I think when people talk about high temperature processing, they often think of vitamin destruction. In fact, fat-soluble vitamins are not affected by the heat treatment. So it's only water-soluble vitamins that are affected. And again, the effect does increase with the severity of heating. UHT milk, the losses are about up to 10% for most of the water-soluble vitamins, except folic acid and vitamin C, where the losses are higher. In container sterilised milk, of course, the losses are much higher, up to 30% for a lot of them, but folic acid, B12 and vitamin C, the losses can be up to 90%. So consequence of that, obviously, is that it reduces the nutritional value of the product, but really only in severely heated products. But the other one I want to mention is that the destruction of thiamine, vitamin B1, is the basis for the chemical index C star, which is often used to measure the chemical effect of heat treatment in UHT milk. A C star of 1 is equivalent to a 3% reduction in thiamine, and that relates to a heat process for 135 degrees for 30.5 seconds or something equivalent. And that's the recommended maximum severity for a UHT process to reduce the amount of heated flavour. Many UHT plants, particularly direct plants, or particularly indirect plants, operate at, with a C star of greater than 1. But a lot of direct plants operate at a C star of much less than 1. And of course, the C star of in container sterilisation is much higher, something around 6. So just a few conclusions. Obviously, there's a lot of heat treatments used in the dairy industry, ranging from 60 degrees up to 150 degrees. In addition to destroying bacteria, they cause numerous chemical changes, including inactivation of enzymes. Some of these changes are beneficial, some are detrimental, but the detrimental effects on nutritive value and safety are really only significant for products with severe heating. So thank you very much for your attention.
Thanks, Hilton. That's really a great overview of what's covered in the bulletin. Appreciate you kicking things off with that fantastic overview. There's been a few questions about whether your presentation is available. I assume you can make that available to participants.
Yeah, I can do that.
Yeah. Is that a yes?
Yes, for sure.
Okay,
yep.
Thanks. And I would encourage all participants to please go to the IDF website and download a complimentary copy of the bulletin, the cover of which is shown behind me on my virtual background. Let's now delve a little deeper into some of the content of the bulletin, and we'll move to Walter Bissig. Walter is a senior research scientist at Agriscope Food Microbial Systems in Bern, Switzerland. Walter specializes in cheese quality and the cheese microbiome. He has a master's in food science and technology and a PhD in food process engineering. He's held a number of leadership positions in both Switzerland and France that have covered new product development, processing technologies, and particularly quality assurance, safety, and compliance. Walter's authored and co-authored more than 80 publications. He works closely with industry to continually improve cheese quality. And of course, part of that drive for improvement is related to heat treatment. His presentation today will cover thermization for cheese milk or before milk cold storage. So over to you, Walter.
Can you hear me now? Okay. Can you hear me?
Yes, it's clear.
Can you hear me?
Yes, well, that's clear.
Can you hear me?
Yes, well,
Yes,
can
yes,
hear me.
yes, yes.
Oh,
Yes, we can hear you, Walter.
yeah, okay. The presentation should start now. Yeah.
There we
Now
go.
it's on the screen, isn't it?
Yes, thank you.
So as Jeff said, I will talk about thermization of cheese milk or of milk before cold storage. The outline of my presentation is that I give first a definition of thermization and legal aspects, if there are some. And then thermization of cheese milk is a focus, give some information about different hurdles in cheese making, the raw milk quality, which is very important for cheese made from thermized milk, the heating conditions applied for cheese milk, and compare cheese with cheese from pasteurized milk. And then thermization before milk storage, how is that done, and some conclusions. Codex Alimentarius is defining thermization in the Code of Hygiene Practice for Milk and Milk Products. And it says thermization is the application of milk of a heat treatment of a lower intensity than pasteurization that aims at reducing the number of microorganisms. And they say that about a reduction of 3 to 4 logs are expected. Of course, that's very much dependent on the bacteria species and also strains. So we can't generalize that. And but also the surviving microorganisms, they are heat stressed, and if a further heat treatment is applied afterwards, they are killed more probably. In Switzerland, we have labeling of cheese, which is made from thermized milk, and it's defined that if milk is heated between 40 degrees and 72 degrees for more than 15 seconds, or at least 15 seconds, then we can label it as a cheese from thermized milk, but it's voluntary, it's not compulsory. Otherwise, yeah, it has to be said that it's made with raw milk at least. But in other countries, there are very few regulations about thermization. It's mostly not defined. And Hilton, in the Encyclopedia of Dairy Science, defined it as sub-pasteurization heat treatments, different kinds, as you already said in the previous talk, Hilton. So first I talk about thermization of cheese milk, the hurdle concept. You probably have heard about this hurdle concept for food safety of cheese. Here for semi-hard cheese, we have this hurdle concept, which was described by Leisner and Corris in 1995 and developed already before by Leisner mainly. And I added this previous step of milk hygiene before these steps mentioned by Leisner and Corris. The milk hygiene is very important for raw milk cheese and as well for cheese made from thermized milk. So we have to have a very low bacteria count in raw milk, lower than 10,000 total plate count, and very fresh milk as well. In this example I mentioned here at the bottom, that the one AOP cheese, the milk has to be less than 24 hours. So we really have to make cheese every day. We can't keep the milk over the weekend because we want to have Sunday or Saturday off. Then we can apply thermization as a step. I mentioned here already these conditions: 65 degrees for 15 seconds, or equivalent 60 degrees 5 minutes, or 57 degrees for 30 minutes. This is our equivalent conditions. Sometimes also at lower temperatures, shorter conditions are applied. There is also a scalding step during semi-hard cheese production for Datum-1 cheese. As the example, it's scalding at 46 to 53 degrees. Then we have these further steps. I don't want to mention all of them. I go to the last step here. We have also as a starter and non-starter lactic acid bacteria in the cheese. They occupy the ecosystem, so that's an important hurdle as well for the food safety. And in this cheese, Datum-1, as the example, ripening has to be at least 75 days. And during this ripening, free fatty acids are formed, peptides are formed, and other metabolites, and they help to reduce pathogenic bacteria and also spoilage bacteria. We can see here for Listeria monocytogenes, 0.5 logs per month, or pathogenic E. coli, 1 log per month, or Staphylococcus aureus, 2 logs per month reduction. And these further steps in between you can read another time. So we look now at the low count of spoilage bacteria and some more information about pathogenic bacteria. In Codex Alimentarius, we have also further requirements for the production of raw milk cheese, and which applies also for cheese from thermized milk equally. We have to know the health status of each animal, each cow. In the case of cow milk, no visible impairment of health of the animal, no infectious disease caused by human pathogens of all the herds used where the milk is used for this cheese, and animals have to be free of tuberculosis and brucellosis. This is all requirements to make raw milk cheese or cheese from thermized milk. If we look at the raw milk quality as an example in Switzerland, yeah, we have these red lines. These are the legal limits in Switzerland: 80,000 for aerobic mesophilic bacteria, maximum 80,000. For cheese milk, for raw milk cheese, we require, most of the companies require less than 10,000, and typically it's about 5,000. Somatic cell count also has to be low. Legal limit is 350,000 per milliliter, and for raw milk cheese, it's typically below 100,000. And then farmers get more money for the milk if they have good milk quality. There are advantages. We looked at how many pathogenic bacteria we have in the raw milk, or also toxin-forming bacteria. We can see at the top Listeria monocytogenes. It's quite low in Switzerland compared to other countries. The main germs which cause problems in Switzerland are pathogenic E. coli, verotoxin producing E. coli, or gigatoxin producing E. coli in the second line. 1.8, we can see here, these cause problems. And then Staphylococcus aureus as well, the third line from the bottom, 8.3%. Have more than 300 colony forming units in the raw milk. And so thermization and quality insurance for raw milk cheese and thermized milk cheese has mainly to deal with these two species of bacteria. Histamine forming lactobacilli I also mentioned here, quite high, and these have to be managed in the raw milk by hygiene of the raw milk. They are not allowed to be in the cheese, otherwise we have problems afterwards, because they survive, of course, in raw milk but also in thermized milk. So there are other measures necessary. And also here at the very bottom, you can read that Mycobacterium and Brucella abortus and Coxiella burnetii, they have largely been eradicated in developed nations. Of course, they still persist in some countries or also re-emerging, and also in developed countries here, in Switzerland or elsewhere. It's very important to always control herd health. Now we look at the thermization conditions for cheese milk. Here at the left, you have this picture of a cheese where thermized milk is used, Tilsiter cheese, Swiss Tilsiter. And the conditions applied are 65 degrees, 15 seconds, as I already mentioned, or equivalent is 60 degrees for 5 minutes, 57 degrees for 30 minutes. Often this thermization is only applied for the evening milk prior to storage overnight, and the morning milk is not thermized, but used as raw milk to make cheese. The primary purpose of the thermization is to reduce the risk of undesired fermentation in semi-hard cheeses. And other purpose is also, of course, to reduce the pathogenic bacteria, to make it, to apply it as a hurdle for food safety. Advantages compared to pasteurization of cheese milk are that enzymes such as lipoprotein lipase are still, and catepsin, this is mostly still active. Lipoprotein lipase about 50% active. And also thermoduric desired bacteria such as Pediococcus, they are less inactivated during this heat treatment. And diverse lactic acid bacteria and non-starter lactic acid bacteria, they still can contribute to ripening flavor and texture of this cheese. Thermization is also applied to reduce protein loss when the milk is stored for some time, because during cold storage, beta-casein migrates out of the micelles, and thermization reverses this process. We have then, if we use sub-pasteurization heat treatment or completely raw milk, we have a higher microbial diversity, and this contributes also to gut health. We can see here in blue the thermization part, conditions, and the reductions of the different bacteria. Spoilage bacteria mainly are here mentioned. Enterococci, different ones, propionic acid bacteria, they also have to be managed by raw milk quality. They are not enough reduced by thermization. Lactobacillus parabuchneri, or now they are called Lactobacillus parabuchneri, also raw milk management, quality management has to be applied there. Escherichia coli, they are quite well reduced with thermization, and that's also one reason it's applied. We can see these lines of 1 log reduction of the different bacteria I mentioned. Now to the pathogens, how much are they reduced during thermization? We have Listeria, Monocytogenes. They are quite heat resistant in comparison. With 65 degrees, 15 seconds, they are reduced by 0.7 logs, 80%. And then the other ones you can see Staphylococci, the other one I mentioned, which is causing problems, 0.9 logs, or Shigatoxin producing E. coli, 4.4 logs. Yeah, so they are quite well reduced, E. coli, and others less. And so we have to more manage also the raw milk quality again. As I mentioned already, also for Lactobacilli, we have to manage the raw milk quality. During ripening, we have also these reductions I mentioned with the hurdle concept. And because of that, in many countries, especially the US, we have to have a ripening for at least 60 days when cheese is made from raw milk or thermized milk. So because we have also these reductions during ripening. There is a advantage in raw milk cheese quality. We have more proteolysis. Primary proteolysis is about the same, but secondary proteolysis is higher in thermized milk compared to pasteurized milk, as we can see in this orange line. It's higher than for pasteurized milk at the bottom, the red line. Peptidase is originating from indigenous milk microflora, or also proteinases from the milk are still active. We heard that we have more than 60 indigenous enzymes in the milk, so many of them are still active. Also, especially active catepsin D contributes to proteolysis and then flavor development in thermized milk cheese. Lipolysis, we can see here, again, the orange line is thermized milk in the middle between pasteurized and raw milk cheese. Lipoprotein lipase, we have heard, is about 50% reduced by thermization, so it's still active. This is the example of cheddar. Cheddar has quite a low pH also during ripening compared to, for instance, smear ripened cheese. We have a deacidification in smear ripened cheese, so pH is higher also. Lipolysis, also lipoprotein lipase, is still more active and can contribute to flavor development during ripening. Then for milk cold storage, thermization before milk cold storage, and then further processing often applied, for instance, for UHT milk or other milk products. Again, initial milk quality is very important. It is raw milk quality is highly correlated with bacteria count after the thermization and 3 days storage. So milk quality is also important even when we apply thermization. The purpose in this case is to extend the keeping quality of the milk, of the raw milk. It can be extended by about 3 to 4 days before further processing. Especially if we want to reduce psychotrophic bacteria because they multiply during cold storage and they form enzymes which are heat resistant during UHT treatment or other products with a long shelf life, cheese sometimes as well. But I was working in a cheese factory. We used thermized milk and it was not good at all. We had to change the process and use not thermized milk, but directly delivered milk and process it. Thermization reduces the number of spoilage bacteria again here, and also especially the heat labile psychotrophic microflora, Pseudomonas especially, and as they multiply at low temperature. Typically 62 to 68 degrees for 15 seconds is practiced to have this shelf life extension of 3 to 4 days. The reduction, we can see here, reductions of total plate count about 2 logs or a bit less, coliforms 1 to 2 logs, and especially the psychotrophs we want to reduce, and we can reach that by 3 log reductions. And that's the main problem. The main problem are the psychotrophs, and we can reduce them quite well. So the purpose is reached somehow. It's again important to mention that this milk is not safe for direct consumption. Some conclusions, yeah, we can see for raw milk cheese and cheese from thermized milk, it has a superior quality. Also for the milk quality is extremely important. Yeah, the germs which are there at the beginning, we only reduce them but can't eliminate them. So milk quality is important. Thermization can contribute to cheese safety as one of the hurdles, as we have seen, especially for semi-hard cheese. Hard cheese are safe anyway without thermization, but for semi-hard cheese, it can be applied as a hurdle, and sometimes also soft cheeses, but still then every cheese lot has to be tested. Cheese made from thermized milk compared to the ones from pasteurized milk have more proteolysis, a tendency of more lipolysis as well, and this improves cheese flavor, texture, and characteristics, and also microbial diversity for gut health is higher. Thermization of milk before cold storage and further processing allows 3 to 4 days shelf life extension at 4 to 6 degrees. And bacteria count of thermized milk after cold storage is directly correlated to the bacteria count before. So again, milk quality is important. That's what I wanted to say, and we can have questions later on. Thank you.
Thanks, Walter. Appreciate that very comprehensive overview of thermization. We do have a number of questions coming in from participants. Please keep them coming. We'll turn to a Q&A session after all the presentations. Our next presenter is Kieran Jordan. Kieran worked in microbial food safety, including with Bacillus cereus within the food safety department at Chaggas in Moore Park Island. Although he's now retired, Kieran has continued his work with the International Dairy Federation. He's a member of the Standing Committee on Microbiological Hygiene and a member of the Science and Program Coordination Committee. Kieran is joining us today to address the topic of challenges with Bacillus cereus in extended shelf life ESL milk. Over to you. Welcome, and over to you, Kieran. Kieran, you might have to unmute.
Sorry about that, folks. I was on mute and I forgot to unmute before
Thanks.
presentation.
All good now.
So my presentation, good morning everybody, you're all very welcome to the webinar, and my contribution is on extended shelf life milk, and the extension of shelf life through heat treatment varies from 15 to 60 days, which is quite a large variation, but that variation depends on the time temperature combination used, on the raw material quality, that is the milk quality coming into the factory, and on packaging, sterile packaging, and other issues as well. But the main issues would be raw material quality and time temperature combination with regards to Bacillus cereus. As I said, and as others have said this morning, there's no standard specification for the heat treatment of the milk for ESL milk. It varies from 120 degrees to 140 degrees to for 1 to 10 seconds. And that's quite a huge variation, and that's why you get such difference in the length of time for the extension of milk shelf life. The process can impact on the flavor of the milk, as Hilton had mentioned earlier about protein interactions and Maillard reaction and that. So in general, and I'm only saying this in general, the higher heat treatments for the shorter time have less of an impact. So that the impact on flavor really has to do with time as much as temperature. The process for the production of ESL milk will kill all vegetative cells of bacteria, that is free-living bacteria and non-spore forming bacteria, and it will also kill some spores in the process, and we'll come to that later. Just a small bit about spores for those that are not entirely familiar. Some bacteria can form spores when they sense unfavorable conditions in their environment. It's really a survival mechanism where the DNA is protected by a strong outer coat, which is resistant to heat treatment, to cold, other treatments like acid or that, and spores can survive for hundreds of years without being affected, and then after that they can be revived again. So they really are a strong survival mechanism that are used by some bacteria. And Bacillus cereus is one of those bacteria that can form spores and therefore can survive the various heat treatments in ESL milk. Not all Bacillus cereus can survive the heat treatment. It's dependent on strains. Some strains can survive. Their spores are strong enough to survive, but others don't form that strong a spore and will be inactivated by the heat treatment of the ESL production process. Bacillus cereus, we generally refer to and imply that Bacillus cereus is an actual bacteria, and it is, but there are also several other bacteria in the Bacillus cereus group that are very difficult to distinguish between Bacillus cereus and Bacillus mycoides, for example. And the members of the group, maybe 6 or 8 different members of the group, are referred to as Bacillus cereus, even though they might not be Bacillus cereus itself. So the differentiation of Bacillus cereus, the actual strain or species Bacillus cereus, I won't go into that at all. Just say that be aware that when somebody refers to Bacillus cereus, they might really mean Bacillus cereus group, and other members of the Bacillus cereus group are not as, if you like, pathogenic as Bacillus cereus itself. That's apart now from Bacillus anthracis, which is causes anthrax and is in the group quite a serious organism, but it's not associated with milk. The Bacillus cereus is the main toxin producing, apart from anthracis, the main toxin producing strain or species, I should say, in the group, and some strains of Bacillus cereus produce toxin and are psychotropic, and they really in ESL milk are the important strains, the ones that can grow at 4 degrees and they also produce toxin. Ones that grow at 37 degrees and produce toxin are not as important as the psychotropic strains, because their milk would be stored at 4 degrees rather than at 37. So at 37, the spores would remain dormant, whereas at 4 degrees there can be growth of the strains and toxin production. So with Bacillus cereus, toxin production can occur before heat treatment, sterilized toxin involved, and that can be produced in the raw milk before the heat treatment. And then while the spores might be killed during the ESL process, the toxin itself will survive the process and can cause illness. And that's an emetic toxin, which causes vomiting. When you get Bacillus cereus surviving the heat treatment and growing in the milk, that would produce diarrheal toxins. So you've got different types of toxins in the related to Bacillus cereus in the ESL milk, and I'm not going to go into too much into that differentiation, except to know that there are toxins produced in the raw material and then surviving the heat treatment, or the spores surviving the heat treatment and producing toxin afterwards. And they're really the two main issues with Bacillus cereus in ESL milk, and the two different kind of toxins that are produced. So contamination, as I said, can occur with toxin before the heat treatment, and that's a very different thing, and a very different remedial action that's required if that's the problem. You can have contamination with spores of toxin-forming psychotrophic strains. The spores survive the heat treatment and then grow at 4 degrees and produce toxin. Or you can get contamination during production. And the particular point in production where contamination occurs, according to a lot of research, would be in the biofilm in the filler of the treated milk. And this can be shown by source tracing of the isolates from finished product, from the processing and from the raw materials. And that will be done using modern genetic methods where you can show the same strain isolated from the filler as is in the finished milk, for example. And that is evidence that there was contamination, or it's normally through biofilm, the contamination at that stage on the filler, and that as I say, can be demonstrated with genetic methods. I'm not really going to go into all the occasions on which Bacillus cereus has caused illness and how it has done that and all that, except to say that it can happen to you. There's a general tendency out there to think that, oh, other people have problems with Bacillus cereus, but we don't. And I want to dispel that myth and say it can happen to you. But the important thing to remember is that it can be prevented if you have good quality milk. And all speakers have been over this already. You know, good quality raw milk is the answer to an awful lot of problems. Hygiene during the processing, attention to detail in cleaning and hygiene can address and preempt an awful lot of problems, and particularly with the filler, a lot of work has shown contamination with biofilm of Bacillus cereus on the filler. If your hygiene process is good enough to prevent biofilm and the attention to detail is on the filler, then you have preempted a lot of problems. Packaging, of course, always helps. And another thing that helps a lot is just awareness, to be aware that there can be issues with raw material, or there can be issues with processing, and that awareness. And as I say at the start, the awareness that it can happen to you. It's not always others that are affected by this. And all of these issues of raw material quality and attention to detail and cleaning, these are all within the capability of management of a processing facility. It's not rocket science to have good quality raw material, which should be a primary objective, and it's not rocket science to have good cleaning and hygiene practices. And there are low costs in these kind of issues with good quality raw material, cleaning, awareness. There might be some costs in packaging materials and all that, and that, but that's where risk-benefit analysis comes into it. But the primary thing is that preventing Bacillus cereus in ESL milk is within the capability of management of a facility and doesn't cost an awful lot. That's about all I have to say this morning on ESL milk and Bacillus cereus. Any questions? No problem. As I say, I've kind of summarized it fairly concisely and said something in one sentence that could really address the whole presentation, but there isn't time for that kind of detail. So if there's any more detailed information that anybody wants, my email address is there. You can contact me by email, or I can address some questions at the end of the webinar. Thank you very much.
Thanks, Kieran. That was really great overview. And as Kieran says, if there are any specific questions, please put them in the chat box and we'll try and get to them following the final presentation that's coming up. Our next speaker and our final speaker is Jonas Eden. Jonas is a technology specialist at Tetra Pak Processing Equipment in Sweden. He has more than 15 years of experience both at Tetra Pak and earlier in R&D positions at a dairy producer and manufacturer. He has a master's in biotechnology and is a licensed engineer in food technology and engineering, both qualifications gained at Lund University in Sweden. Today, Jonas is going to talk about technologies for heat treatment, design of heating systems to achieve expected shelf life and product quality. Over to you, Jonas.
Thank you. Hope you all can hear me.
Yes, we can.
Yep, perfect. So my name is Jonas Eden, and I'm from Tetra Pak Processing Equipment, and I will talk a little bit about heating technologies, a little bit about the design and so on from both ESL and UHT. So if we start off, we look at the key technologies, and we start off from the left. We have pasteurization. Just let me, so I can see here. To the left with pasteurization, where we have the lowest heat load on the milk, but we also have the shortest shelf life, which you can see in the graph at the bottom. You can see shelf life. Typically in Europe at 6 degrees, you have 7 to 8 days shelf life, but this depends on several factors, as we have discussed, like raw milk quality and so on. And in combination with pasteurization, you can apply technologies before the pasteurization, which is Bacterfuge, where some of the spores, like Bacillus cereus, which we have discussed, that can influence shelf life, can be reduced by 1 or 2 log, which adds, depending on several factors as well, up to 7 days of shelf life. And it has no increase on the heat load on the milk, so it's still pasteurization. And then you can also use microfiltration, which not only reduces the spores like Bacillus cereus, depending on pore size, it's between 4 to 6 log. But it also reduces the number of vegetative thermoduric psychotrophic bacteria, which are the ones that can survive pasteurization and can grow in cold storage. They do not grow as fast as Bacillus cereus, but over time, they can still grow and spoil the product or be in very high total plate count. But with microfiltration, you can get up to approximately 40 days shelf life. However, when you use microfiltration, you cannot microfiltrate the cream part of the milk. That has to be high heat treated separately and then sort of standardized back into the milk. So you will have some increase in heat load on the milk. If you want to have a longer shelf life than 40 days, both due to the bacteriological flora in the milk, but also enzyme inactivation and so on, you will have to go on to high heat treatment. High heat treatment for ESL or UHT heat treatment are the same, and the unit can be designed to do both. And the high heat treatment can be either indirect using tubular plate heat exchanger or direct with steam injection or infusion. And as you see in the graph, you have the time versus temperature, and you can see you have pasteurization, the green as a reference, and then you have the dark blue, which is indirect. You can see it quite, there is an incline, and compared to the direct, it's a slow incline. For the direct, it's very almost instant heating from 80 degrees up to UHT temperature in this case. And for the indirect, it takes quite a long time to get up to UHT temperatures. And also during cooling, you have the flash cooling, which is very instant, where when you remove water from the product which you have added through the steam. And the indirect, you have the heat exchanger, which then takes a certain amount of time. So the very rapid heating, as you can see here, with the injection, where it injects steam into the milk, the red is the steam, or using infusion, where you have steam in a chamber, and then you have milk coming from the top in droplets pouring down, and then you have the exit in the bottom. Both of these result in minimum heat load on the product and also the highest quality of the product. But it's not only the technology that can influence the heat load or quality of the product. In order to provide the highest product quality, the program, Tetra Pak program, automatically, if you have sort of capacity change resulting in, so you go down to half capacity, that will mean that you have a longer holding cell, meaning that you will over-treat your milk. And by calculating F0 or F value, depending on if it's UHT or ESL, you can ensure by regulating the temperature from time that you have the highest quality, but also food safety. So the F0 control we have just discussed, but there are other ways you can also minimize heat load if you have variable capacities in the line. One is by having a split heater. This means that you can see in the figure to the bottom right, you see that the final heating is split into two heating sections. And this means if you go from full capacity down to the half capacity, as you see in the graph, where you have time and temperature, the first section in the final heater there the water can be bypassed, meaning that the heating is delayed until the second section, and then you have the heating in the second section when you have the half capacity. That means that the area under the dotted line is removed from if you would have one heating section instead, meaning that you will have, even though you go down to half capacity, you still have a very high quality product compared to if you would have one section. But of course, if you know that you're running half capacity from start during the whole production, you can manually remove one section, meaning that you will reduce the surface area of the heat exchanger. For indirect UHT or ESL, optimizing the hot water flow is also very important. This is not to have that the product reaches final temperature early in the heat exchanger. You want it to reach final temperature as late as possible in the heat exchanger, so that you don't over-treat the product more than necessary. So optimizing the hot water flow depending on the heat exchanger is very important for the quality and the heat load as well. And I just want to mention as well, during the design of the heat exchanger, we look at different types of heat load indicators and try to calculate this and try to predict the heat load of the units so they are within requirements and so on. So as you see on the graph, you have two heating units for ESL, general heating units. One to the left, which is a direct, and one to the right is an indirect, which is including a protein denaturation holding cell or protein stabilizing holding cell. And as seen in both of them, you can see protein denaturation and furosin formation. Of course, furosin, as mentioned, is not formed in the milk. It is when you analyze the milk for lysine lactose reaction, you add acid and heat, and then it's formed into furosin. So it's the furosin that we measure during the analysis, but it's not formed, as mentioned. But as you can see, for an indirect heating, it's much higher, and the denaturation is much higher as well compared to a direct heating unit where you have a very instant heating and cooling. And also I'd like to mention, very important as well for the design is the energy recovery. And by using an indirect heating with product regeneration, the energy consumption can be highly improved as well. And the final design I like to mention as well that you can do to improve the product quality is if you look at the traditional UHT or ESL line, with you sort of separate, standardize, and pasteurize the milk, and then you store it before the UHT or ESL production. But by using a one-step technology, you sort of remove the pasteurization and the mid-storage tanks, and you go directly from raw milk, separation, standardization, then to UHT and ESL heat treatment directly, and then to filling, meaning that not only do you minimize the heat load of the product, but you also reduce energy consumption, water usage, footprint, and etc. So that was a short presentation for me. Thank you.
Thank you, Jonas. Very good overview of the technologies available. We might now turn, and before I do turn to questions, I want to thank all the speakers. And if we don't get to a question, one of your questions for a particular speaker, please follow up with them directly after the webinar, or you can certainly contact them through IDF. So let's turn to some of the questions, try and cover as many as we can. The first one I think is probably for Walter. Does thermization cause damage to milk, and how much does it reduce spore-forming microorganisms?
Yes, spore-forming microorganisms, they are not reduced. I mean, at least the spores are not reduced. So these spores are still there, they are not affected. And then chemical change, as Hilton said, there is practically no chemical change. It's really, even lower than in pasteurization. But with pasteurization, it's low already. I think we have about, maybe you often use beta-lactocobalin denaturation as indicator. For pasteurization, it's about 20%, I think. And for thermization, it's even lower. So there is very little change. Some change, as we saw, lipoprotein lipase is reduced by 50%, about 50%. So there is some change, but very, very little.
Okay, thanks, Walter. We've also had some people requesting a copy, I assume a PDF copy of your presentation. Would that be okay?
Yeah, PDF is okay, yes.
Okay, thank you. The next question I think is probably for Hilton. What is the best index to measure heat damage to milk during concentration? You need to go off mute, Hilton.
Yeah, thanks, Jeff. I haven't seen anything on heat indices for concentrated milk, so I'm assuming that lactulose would still be the best index, but I don't have any proof of that.
Okay, thank you. Next question, I guess, is to any of our speakers. It's a question about what influences the behaviour of milk in foaming applications, and it's particularly related to foaming for various coffees, cappuccinos, things like that. I guess that's open to any of the speakers to address what are the main factors that influence foaming behaviour.
Well, if I could kick it off, Jeff, one of the main factors that decreases foaming is lipolysis and production of free fatty acids, and the other byproducts of that are the partial glycerides, which have surface active properties. So high free fatty acid levels in milk certainly depress foaming activity.
Thanks, Hilton. Any other speakers have further comments?
Yes, I fully agree with that. And yeah, recently we also saw that, for instance, dairies made experience that milk processed on Monday has less foaming activities than milk processed on Thursday or so. So really, proteolysis by enzymes from psychotropic bacteria, they also can influence foaming properties, and that's also research in Norway done, which says this. Yeah, Sif is probably here, and she, as a participant, and yeah, she has this experience on her colleagues.
Thanks, Walter. Another one probably for you, Hilton, about lipoprotein lipase. It's a question about what heating conditions are known to be best to avoid any potential lipase activity in casein concentrates, and what consequences can be expected regarding renotability of those casein concentrates.
I'll just talk about the lipoprotein lipase one. Someone else might like to pick up on the renotability. The lipoprotein lipase is inactivated by normal pasteurization conditions. I don't have information on the effect in concentrated products, but I'm assuming that it's going to be very much the same as for milk.
Maybe Kieran, can you comment on the renotability question?
Fred Nash.
Or maybe Walter.
Microbiology is my area of knowledge, and limited knowledge on other issues like that. Sorry.
Maybe Walter.
Renotability about concentrates, dried concentrates. I'm not a specialist, but yeah, I know that sometimes native casein, micellar casein is used to make cheese. But this has to be treated very low temperatures, and so renotability is still there. But I don't know about casein, what type of casein. It depends, of course, a lot on that. It has to be micellar casein. There are some powders like this, but as I said, they have to be treated very carefully, very at low temperature as much as possible.
Okay, thanks, Walter. A question about an analytical method to check for raw milk and whether raw milk has been pasteurized, particularly in relation to ovine milk. Can anyone comment on the best analytical method to check if that raw milk has in fact been pasteurized or not? Hilton?
Maybe Hilton. No, not really, Jeff. We've seen some of the changes that occur with
alkaline phosphatase, couldn't you?
Yes, well, that's right. We've seen some of the changes that occurred during pasteurization. So I guess if we reverse that, we can certainly tell if we've got raw milk there. So alkaline phosphatase is probably the most obvious one.
Yes, yeah, I would have thought so. One for you, Walter. It's about the release of beta-casein from the micelle during cold storage. You mentioned in your presentation that thermization can reverse that. The question is related to kinetics. It takes a relatively long time for the beta-casein to leave the micelle during cold storage, but is the reversal when you heat a much more rapid process?
Yeah, to my knowledge, this thermization has this effect. It's applied for this as well. And yeah, thermization doesn't take long, as we said, 65 degrees, 15 seconds, or 60 degrees, 5 minutes as an equivalent. So it goes relatively quickly, I presume, yeah, as this is observed during thermization. So it must happen during this time. But okay, I'm not a specialist on this kinetics. Yeah.
So the reversal is much faster than the release.
Yes, I would say so. Yes.
A question about ESL versus sterilized milk. What is the difference between ESL and sterilized milk? Maybe that's Hilton again. I think you covered that in one of your slides.
I may have, yeah. Sterilized milk contains virtually no bacteria. UHT sterilized milk will contain some very heat-resistant spores, but in container sterilized milk will have virtually no bacteria. ESL milk will have some bacteria, as we've heard. It's not heated to a degree that inactivates all the spores, and so there'll be some spores remaining in there. Neither has any vegetative cells, but some spores will remain in ESL.
Thanks, Hilton. One for Jonas here. What's the minimum capacity Bacterfuge available from Tetra Pak?
So I don't know exactly the minimum capacity, but if they send me an email, I can guide them to a person that can answer that question. I mean, we have a minimum like nominal capacity, but you can always go down as well. So I cannot answer that directly. So please send an email and then I can answer, or send them to somebody that can answer.
Arnett Dautre, maybe if that participant is still on the webinar, they can contact Jonas directly. We haven't got much more time. We'll finish up shortly. A couple more questions. Dealing with vegetative bacteria in milk or whey, what is the best choice between microfiltration and Bacterfuges? Maybe to you, Jonas. So they're looking for pros and cons between the use of microfiltration and Bacterfuge to remove vegetative bacteria.
I mean, a Bacterfuge cannot take vegetative bacteria. It can only take spores because the density of them is similar to proteins. So they cannot remove it. But the filtration can remove vegetative cells as well. And then depending on which type of vegetative cells, I mean, you have very small ones like micrococci and so on that are harder to remove than larger ones. But the main difference, pros and cons, is that the filtration can remove them and the bacterification sort of cannot.
Okay.
So you will get extended shelf life of that as well.
That was from Jose Toro. If he wants more information, perhaps contact Jonas directly.
Yes, please.
Maybe a final question. How is lactulose measured? Perhaps for you, Hilton?
Well, it's normally measured by GC, gas chromatography. There are other methods available, but that's probably the most common one. There are enzymic methods available as well. So there are quite a few methods around.
Okay, thanks, Hilton. It's coming up to nearly 90 minutes since we started the webinar, so we might start to finish up now. It just leaves me to, first of all, thank our 4 speakers, who I think did a great job in providing a good overview of the material available in the bulletin, the cover of which is behind me there on the virtual background I'm using. Please go to the IDF website homepage and navigate to publications, and you'll be able to download a PDF of the bulletin free of charge. I also want to thank all the participants. At some stage we had well over 200 joining this webinar today, which is really a great turnout. Thank you for participating. Thank you for your questions. If we didn't get to your question, or you have questions after the webinar finishes, please contact our speakers either directly or through IDF. They can put you in charge with the right person. Okay, we'll finish up there. Thanks again, and bye for now. You there, Aurelie?
Yes, I was trying to mute the noise.
Okay. We're getting lots of thanks from people. I think everyone enjoyed it. And thanks again, Hilton, Walter, Kieran, and Jonas.
Okay, thanks, Jeff.
You're welcome.
Thank you. You're welcome.
Yes, thank you, Jeff, for the good moderation of this seminar.
Yeah, you're welcome. I think it went very well. We're getting a lot of compliments, so it was well received.
Okay.
Thanks.
Yeah, thank you.
Thanks very
Thank
much,
you all for
folks.
putting this together.
All right.
Bye now.
Okay, bye.
Goodbye.
Thank you.
Bye.
Thank you.
Thank you.